Hi, I'm Deborah Meyers from Wake Forest. And this is Kelan. You want to introduce yourself?
Oh, sure. I'm Kelan Tantisira. I'm from the Brigham and Women's Hospital.
So today we're discussing your recent paper on steroid response, especially in children. Do you want to just summarize the major findings?
Oh, sure. So in this particular study, this was a genetic study where we evaluated genetic response to clinical symptoms in asthma in response to inhaled corticosteroids. And we thought that, of the three major domains of impairment, being lung function, exacerbations, and symptoms was one domain that has been largely untouched in the asthma literature and in terms of genetic association in particular. And so in this pharmacogenetic association, we evaluated children from the Childhood Asthma Management Program as a primary cohort, did a genome-wide association study, and then evaluated for replication several additional cohorts, one which was a childhood cohort from the CARE network, and then two adult cohorts from the ACRN and the LOX trial, respectively. Globally, what we found was that there were several variants that after we evaluated in combining the two pediatric population, there were three variants that approached, actually met genome-wide significance, and of which one was particularly interesting because it mapped to a gene. The other two were intragenic and mapped to an expressed sequence tag, respectively. And so the rest of the manuscript actually focused on the variant that mapped to the gene, and it was the FBXL7 gene, which I believe stands for FBOX of lysine protein associated number 7.
Would it be a gene we would have picked?
It would not have been a gene that any of us would have ever picked. And the interesting thing about the association was that while it met conventional genome-wide levels of significance when combined into two pediatric populations, it was really a very specific pediatric association, and the association did not further replicate in either of the adult populations. Now, there have been other asthma studies, asthma GWAS studies, where loci have been identified that were just uniquely pediatric specific or uniquely adult specific. And obviously, from a symptoms perspective, it may be that kids perceive, and the mechanisms for childhood perception of asthma severity may be different from adult severity as well. The final thing that we did was, the final two things we did in the paper was, one is we looked to see if there was any evidence that the particular SNP was functionally relevant. And in order to do that, we used an expression quantitative trait loci cellular data set where we stimulated lymphoblastoid cell lines derived from the same childhood asthma management program, individuals with dexamethasone, and indeed found that the SNP of interest was also associated with gene expression, suggesting the potential for functional likelihood, although further work would need to be done to prove that. The final thing that we did was we said, well, how might other genes be influenced, or how might FBXL7 be interacting with other genes? By looking at a pathway analysis, we noted that FBXL7 does seem to interact with another gene, which is HIF1A. And that gene, while not specifically associated with asthma before, has been extensively described in neuropathic pain. And so theoretically associated with quote-unquote perception per se, and perception of pain specifically. So maybe another mechanism specifically with how people perceive symptomatology as compared to just pain.
Were you surprised that the adults didn't replicate it? Although, like you said, that's been seen in other studies also.
Well, obviously, the best genetic studies are those that replicate everywhere you try, because they're certainly the most ubiquitous. But I do believe that the adult asthma phenotype, even independent of kids, does seem to be different.
I would
both
agree with that.
Clinically, physiologically, and otherwise as well. So it certainly is understandable that there may be some differences.
Just the fact that the adults have been on steroids probably for many years and the children are basically naive to steroids.
Absolutely,
right?
that might be the case as well. So.
And plus the difference in symptoms for children, of course, lung function may not be a good predictor because it's basically normal in many asthmatic children.
Exactly.
Symptoms are going to be much more useful.
Right.
Yes. So are you carrying this work any future directions on comment?
Well, I think that certainly we are slowly but surely developing our functional laboratory, and so certainly we're interested in pursuing anything that looks functionally relevant and interesting, and so we can consider doing things such as siRNA experiments to see if there's any differences in expression by inhibiting the FBXL7 gene, and beyond that we also have other genome-wide association analyses ongoing to basically look at slight modifications in how you define symptoms and to see if there might be other genetic loci that may either be adult-specific in terms of their perception or just based on having more of a more severe symptom phenotype, whether there's something that is geared towards those ones.
So do you think maybe you'll be able to develop, for example, like a genetic profile to help individuals as far as how responsive they are to certain therapies such as steroids?
Well, so I think that certainly that is the ultimate goal of all pharmacogenetics. Gain, on one hand, you gain biologic knowledge, but on the second hand, ultimately, those of us, yourself, myself, who are in the field of asthma pharmacogenetics, our goal is really to try and to help with the individualized medicine approach, to try and tailor appropriate therapy and identify a priori, hopefully, individuals who will and will not respond to therapy.